Hello, everybody. My name is Susana Kimura-Hara. I'm an assistant professor at the Department of Chemistry at the University of Calgary. I would like to thank everybody for coming today to my presentation. Today, I will be talking about a novel analytical method for the quantification of disinfection byproducts at trace levels in recycled wastewaters. But first, I would like to acknowledge the students and staff that worked on this project. In this case, Alejandro Ortega Hernandez, Rafaela Caballa, Chad Verwold, and Jill Murakami. So water disinfection is the worldwide commonly used practice that inactivate pathogens and provide safe drinking water. Commonly used disinfectants include free chlorine, combined chlorine or chloramines, ozone, and chlorine dioxide. Ever since the first continuous chlorination of municipal water was used in 1908 in Jersey City, a steady decline of waterborne diseases such as typhoid fever and cholera has been observed. And so by the 1940s, the vast majority of water supplies in developed countries had complete treatment and were considered microbiologically safe. And so in this graph, we have the death rate per 100,000 population due to infectious diseases on the y-axis, and over the years from 1900 to 2000. And as you can observe, there has been a steady decline due to chlorination and other medical breakthroughs. This reminds us of the importance of water disinfection and a common practice that is currently being used today. However, these chemical disinfectants may react with natural organic matter and other ions that are naturally occurring in river and lake waters. And so these chemical disinfectants react with these other compounds to unintentionally form disinfection byproducts. To date, there's more than 700 DBPs or disinfection byproducts that have already been identified. And some of them pose a health risk as indicated by some epidemiological studies. Certain chemical disinfectants can promote the formation of different disinfection byproducts. Currently, the US and Canada have maximum contaminant levels for several disinfection byproducts, including trihalomethanes and haloacetic acids, which are predominantly formed by chlorine disinfection, bromate, which is predominantly formed by ozone disinfection, and chlorite, which is predominantly formed by chlorine dioxide. While only certain states in the United States have guidelines for nitrosodimethylamine or NDMA, NDMA is not currently regulated in the US. However, in Canada, NDMA has the maximum concentration guideline of 40 nanograms per liter. So as you can see in this table, there's different disinfectants on the left-hand side, and they can form different regulated and non-regulated disinfection byproducts. So some of them I already mentioned. Chlorine predominantly forms trihalomethanes and haloacetic acids. Combined chlorine forms NDMA, but it's not currently regulated in the US. But there's a guideline in Canada, and chlorine dioxide and ozone form chlorite and bromate, as mentioned. However, these disinfectants can also form other types of disinfection byproducts. For example, combined chlorine can promote the formation of nitrogen-containing compounds, including NDMA or nitrosamines, cyanogen halides, halacetonitriles, and halacetamides. Other classes of concern are iodinated disinfection byproducts, including iodotrihalomethanes and iodoacids. So why are we concerned with these unregulated disinfection byproducts? Dr. Pleva from the University of Illinois in Urbana-Champaign has tested more than 100 disinfection byproducts with one single bioassay. What this allows is that we can directly compare the toxicity of different disinfection byproducts. And so in this graph, as you can see, you have the different disinfection byproduct chemical classes. We have the 5 salicylic acids that are regulated, as well as the non-regulated, nitrogen-containing non-regulated disinfection byproducts. And so in this graph, it shows that it directly compares the cyto and genotoxicity of these compounds. Cytotoxicity means the concentration of a compound that produces cell death, in Chinese hamster ovary cells in this case, and genotoxicity is the concentration of a compound that produces DNA damage. And so the larger the graph, the larger the bar graph, the more toxic it is. And so in this in vitro studies, they've shown that the non-regulated halocitonitrile amides and nitromethanes are more cyto and genotoxic that are currently regulated. Another thing that they found was that the brominated and iodinated DBPs are more toxic than the chlorinated disinfection byproducts. Additionally, but what does it mean for human health? So an epidemiologic study found a higher increased risk in the incidence of bladder cancer with the consumption of chlorinated surface water. There's another study that has also found associations between disinfection byproducts and adverse reproductive outcomes, including low birth weight and preterm delivery. And so although there are risks to long-term exposure of disinfection byproducts, the whole reason and what we must remember is that we apply water disinfection to protect human population from the acute risk of pathogens that produces waterborne diseases. So even though there's chronic risk, we must remind ourselves of the acute risk and that we don't want to catch for waterborne diseases. So with an evolving environment, however, with an evolving environment, that is an increase in population, climate change, and as well as older infrastructure for wastewater facilities, these can lead to higher amounts of compounds into lakes and rivers that are human-made or man-made. And so one example is treated wastewater that are being introduced into lakes and rivers that are then used later on for drinking water purposes. And this we call de facto wastewater reuse. So de facto wastewater reuse is the unintentional use of wastewater to drinking water sources. For example, in this graph, we can observe that City 1 obtains their source waters from a reservoir that's fed by snowpacks and glaciers in the mountains. They're coming down the mountains, similar to the city I currently live in, Calgary. Calgary then uses this water and then produces wastewater, which is treated and then returned to the river. This river will then flow to City 3, and City 3 then uses water from that particular river for drinking water. Therefore, there's a percentage of that river that has treated wastewater, and we know this as a de facto wastewater reuse. It's an unintentional use of wastewater. And so there was a study done in the USA in 25 cities and over a 30-year period of time. So 17 of those 25 cities had an increased de facto reuse, about 68%. The 4 largest cities have a de facto reuse about 8% or higher. That means that 8% of their drinking water source comes from wastewater inputs. Furthermore, there's been also wastewater spills across developed countries, including Canada. So for example, in November 2015, there was about 5 billion liters of raw sewage that was introduced to the Saint Laurent River in Quebec. And so, but this de facto wastewater reuse is not only an issue that pertains to developed countries, but also developing worlds, or more so, it's more it's more severe in developing worlds. Countries like Mexico and Brazil have a high de facto wastewater reuse because they have poor sanitation. And so in this slide, it's a review that compiles the maximum concentration quantified in treated drinking waters in Brazil. And so what they found is different emerging contaminants. The highest one are disinfection byproducts, which is expected. However, they also found caffeine, about 5.8 micrograms per liter, and they also found benzoylecgonine, which is the main metabolite of cocaine, at about 652 nanograms per liter. So this is telling us, because it has caffeine and metabolites in the drinking water, that there's a significant de facto wastewater reuse that is coming into the drinking water. And so now if we have these wastewater impacted waters that are less pristine than before, and it has more human-made contaminants, and we apply disinfectants as well as advanced treatments, if it's something for if it's waters that we want to treat for potable reuse. So this can produce a plethora of other chemicals. So then there's some questions that come up. So what kind of compounds are being formed in this water with these new man-made chemicals? Are there new disinfection byproducts being formed? And are the water quality guidelines adequate for reused water? So for example, we looked at the different disinfection byproducts are being regulated, but are those the same with changing water? So we don't know this. And so this is one of the reasons, and one of the things that my research group are interested is to look into the kind of compounds that are being formed with reused waters. And so in order to do this, we had to develop highly sensitive analytical methods that could look at disinfection byproducts at low concentration, especially since emerging contaminants are typically found in low nanogram per liter. So we needed to develop these highly sensitive analytical methods. And so we developed a method of 25 priority DBPs with the PTV GC tandem mass spec instrument. And PTV is a programmable temperature inlet followed by GC tandem mass spec. And so these 25 priority DBPs include halocyanonitrile. So again, there's a cyanogen functional group attached to this compound, triiodo trihalomethane. So there's an iodide in one of these X's, halonitromethanes, trihaloaldehydes, and haloketones. The other thing that we found challenging with these compounds is that they're light sensitive and also unstable, or they can be very reactive. And so we developed this 25 priority DBPs with a GC tandem mass spec. And then this was followed by an improved liquid-liquid extraction. And then we evaluated this analytical method for matrix effects, especially with secondary treated effluent, as well as the overall performance of the analytical method. And so the first thing that we had to select were the mass spec transitions. So for each DEP, there was precursor ions that were selected based on the base peak. That means the most abundant ion. However, the precursor ion for a few DBPs were selected on the second strongest peak. And the reason was because 3 haloketones had a base peak of 43.1. And so so this would produce a very low product ion. So we cannot quantify at such a low mass to ion ratio. We also found a similar issue with 2 iodinated trihalomethanes, where the base peak led to really poor product ions, especially because these compounds are really, really small. I think the largest would be about 250 Daltons. And so some of these, we have to choose the second strongest peak. In this case, the iodinated trihalomethanes, we have to choose the molecular ion. Then after we chose the MS transitions, we continued to do a collision energy optimization. So first, we started with halocetonitriles. They were manually optimized by incrementing the collision energy applied to each precursor ion. The collision energy was optimized once the maximum abundance was observed for the quantifier and qualifier transitions, as shown in this figure. Once we did this manual, we compared it to the automatic wizard that is available in the instrument, and we found very similar values. So then we went ahead and did an automated collision energy optimization for the rest of the disinfection byproducts. The next step in the GC MSMS method were the time segments and dwell times. So we introduced 4 time segments, and these time segments limits the number of chemical transitions to scan per segment. These time segments ensure a higher sensitivity of the triple quad by reducing the number of chemical transitions to scan per each segment. After we chose the time segments, we went ahead and determined the dwell times. And this refers to the sampling or scanning time spent for each peak during the multiple reaction monitoring. Typically, longer dwell times result in a higher number of ion hits to the detector, resulting in increased sensitivity of the analyte. Also, the peak shape is dependent on these dwell times, as seen as in the equation below. So we determined the dwell time based on the peak width divided by the number of total transitions per segment times 15. 15 is that's the number of points we wanted to have across a peak. And so with this, we had a really nice separation of all of the 25 disinfection byproducts. The red arrows signal the different time segments, and we had a really nice peak shape as well for these disinfection byproducts. So after determining the mass spec parameters, we went ahead and improved the liquid-liquid extraction method. We started with an existing method that uses 100 milliliters of sample with 3 times extraction of 5 mils for a total of 15 milliliters of organic solvent. And then it's flowed down to about 200 microliters. And so we then reduced the sample volume to 50. And as you can see from the graph, these are just 2 DBP chemical classes. There's a significant reduction in percent recovery. So we then went ahead and reduced the final extraction volume to concentrate the analytes in the final extract. And we did see a slightly higher recovery than experiment 2. However, handling the handling of blowing down to 100 microliters was a little bit challenging. So we decided to go back and try to use the final extract about 200 microliters. And so for the last experiment, instead of 50, we went to 10 milliliters with an organic solvent about 9 milliliters total, with 3 times extractions of 3 milliliters for a final extract volume of 200. And we observed similar or higher percent recoveries for some disinfection byproducts. We did observe percent recoveries that were lower for another ones. However, it seemed that experiment 4 provided us with the most optimum value for most disinfection byproducts. And so we went ahead and used 10 milliliters to 200 microliters. So what this meant is we reduced the concentration factor from 500 to 50. And this is also very important because we're looking at these analytes in wastewater effluents. So the less a sample that we extract, we could also reduce the extraction of other compounds that are found in secondary wastewater effluents. And so we also did an optimization of the shake time. So we shaked 10 milliliters in triplicate with spiked analytes for 5, 10, and 15 minutes. And we did not observe significant changes with the shake time, so we decided to use 5 minutes. And so we tested the method's performance and compared it again with the original liquid extraction. So we added 100 nanograms per liter spike of the different disinfection byproduct standards in ultra pure water and compared it to 10 milliliters. And we observed there was a very slight changes. In some cases, it was an increase of percent recovery from 31 to 142%. We also observed that we obtained relative standard deviations less than 10% for the spike recoveries. And we also went ahead and did an instrument position study with 7 replicates at 3 different concentration levels, and we observed a relative standard deviation of less than 3%, except for trichloroacetonitrile. So yeah, also in the percent recovery, we did observe a slightly lower percent recovery for trichloroacetonitrile. However, this has been reported in other methods, and it seems that it's a trouble compound for most extraction methods. And so this is the previous study that we compared. So these are the method detection limits from the previous study, as well as our current study. And so the method detection limits for our method was between 2 and 68.9 nanograms per liter. So, and so this is compared to another method that used 100 mils of sample with a concentration of 500 times. And they used a single quad instrument in order to analyze this disinfection byproduct. So this shows that the method that we developed was able to quantify disinfection byproducts at nanograms per liter or low PPT levels in secondary wastewater effluents. And then we also tested the matrix effect. So this was 5 micrograms per liter spike in pure water, and we did the same for secondary wastewater effluent. And we saw the percent recoveries just slightly lower for secondary wastewater effluent. These was comparable with pure water. The other thing, even though we're quantifying percent recoveries, the calibration curves are made from spiking into ultra pure water and then extracting them. And so even though we do have these low percent recoveries, for example, for trichloroacetonitrile, it still quantifies the amount that we spiked it, because we use spiked samples as our calibration standards. So once we developed this method and validated for DVPs in secondary wastewater effluents, so we tested the method in a full-scale research facility here in Calgary, Alberta, named Aqua, or Advancing Canadian Wastewater Acids. So this is a secondary wastewater. It's a wastewater treatment plant that treats wastewater to produce secondary effluent, and then this is then pulled or transferred to the research facility, which consists of a microfiltration unit of 3 membrane units ran in parallel, and then they're treated either through reverse osmosis or ozone oxidation. And so what we wanted to observe is the efficacy of these advanced treatments of microfiltration, reverse osmosis, and ozone oxidation to remove the precursors to disinfection byproducts. And what I mean by precursors are the things that disinfectants react with in order to form the disinfection byproducts. If we remove those compounds or those precursors, then we're going to produce less disinfection byproducts. And so the way that we did this is through doing formation potential testing with uniform formation conditions. And so we obtained samples from each treatment, and we dosed them with either chlorine or chloramines, or in this case, monochloramine. So we dose them and incubate them for about 24 hours at 20 degrees Celsius at pH of 8. After those 24 hours, we stopped the reaction of the chlorine disinfectant with ascorbic acid. And then we extracted right after with the liquid-liquid extraction, and followed by the GC tandem mass spec method that we developed, And so so I'm going to show you in the next few slides is the DBPs that we quantified before and after disinfection. So in this graph, as you can see, these are the disinfection byproducts quantified in the water samples with no disinfection. So here you can see that halocyanonitrile and then iodomethane were quantified in the wastewater effluents. However, they were reduced in the UF treatment, in the microfiltration treatment. However, something that we did notice is that there was 2 new compounds that appeared in the microfiltration, in this case, chloracetonitrile and dichloronitromethane, that were not in the effluent. And I think we believe it's because the microfiltration units are cleaned every 2 weeks with a highly concentrated chlorine disinfectant. And we believe that they tend to leach out over time. So we probably observed some of these compounds forming from the microfiltration unit itself. Then at the UF ozone treatment, what we observed is that we primarily form halocyanonitrile. So this is about 100 nanograms per liter, or 0.1 micrograms. And this is primarily because of dichloroacetonitrile. And also, we observe, so this is, what we observe, this enhanced formation of dichloroacetonitrile is not something that was observed in other full-scale potable reuse facilities. And so it seems that the halocyanonitrile formation could be unique to the composition of the effluent organic matter here in Calgary. So this is something that has not been observed before. And then also after UF treatment, we did see other disinfection byproducts, including halocyanonitrile, halonitromethane, but the highest one was iodo-THM. And there were studies that have looked at this, and they found that these compounds, the dichloroacetonitrile, dichloronitromethane, and iodo-THMs, are typically not well rejected by RO membranes, and they tend to appear after reverse osmosis treatment. So those were the target DBPs that we quantified, but we also wanted to know what other organic halogen compounds were contained in these waters. And so we also analyze it with a total organic halogen method that can quantify any organic molecule that has a halogen attached to it. And so we do observe that these numbers are much higher. So previously they were in the low nanogram per liter levels, but these are on micrograms per liter levels. So we do see with this non-target analysis that the organic chlorine was significantly high in the effluent and microfiltration, so about 100 to 122 micrograms per liter. But this is then reduced at the ozone level, after ozone treatment, and then it is further reduced with after RO. So ozone and RO removed about 65 to 85% of the total organic chlorine. However, we could see that total organic bromine and total organic iodine were some more or less consistent. Total organic bromine were about 6 micrograms per liter as bromide, and total organic iodine was between 3 and 9 micrograms per liter of iodide. So there seems to be some of these brominated, iodinated DVPs in these processes. However, the UFRO completely eliminated the organic bromine and the organic iodine from these waters. So then we analyzed the DVPs that are formed after adding chlorine to each water sample. And so here we could see we have the effluent and then effluent with chlorine. So we could see that these disinfection byproducts are formed after you add chlorine, and we observed that we have a high formation of primarily halocyanonitrile. So this is almost 18 micrograms per liter of halocyanonitrile. This is composed primarily, again, by dichloroacetylnitrile and bromochloroacetylnitrile. The other thing that we noticed is that the effluent and UF samples have very similar results, and we believe is because before we chlorinated, we filtered the samples with a membrane filter, and so we believe that we might have filtered out things from the effluent, and so after we chlorinated, we observed similar formation of disinfection byproducts. That's why in most of our slides, you're going to see that the effluent and the UF samples after disinfection are going to have very similar results. And so after that, we also observe halocyanonitriles as a predominant DVP class due to, again, dichloroacetylnitrile. But we also observe halonitromethanes as well. So, and this is, so after ozonation, we observed that ozonated reduced the halocyanonitriles by 42%, and this has been corroborated by other studies as well, that they observe a lower formation of halocyanonitriles after ozonation, followed by free chlorine. And then for the last section, the last sample, the UFRO treated sample, we do see a formation of halonitromethanes. We first observe a significant reduction of overall all the disinfection byproducts. And I think we believe that this is due to the removal of the organic carbon content from RO. It removes about 85%. And so if there's less precursors, you're going to see less disinfection byproducts as well. However, again, we do observe within these chemical classes, we do observe nitromethanes, in this case, dichloronitromethane and bromochloronitromethane. And and a few one halocetonitrile and an iodo-THM. And so, okay. And so for the next slide, we did the similar disinfection experiments, but in this case with combined chlorine or monochloramine. And first of all, what I want to highlight is the y-axis. So it's not, so for free chlorine, we produced about a total of 18 micrograms per liter. So combined chlorine, we see a less formation of disinfection byproducts. So we see about 8 micrograms per liter in total. For the secondary effluents and UF treated waters, they exhibit similar speciation trends. However, so we see the halocetonitriles and similar disinfection byproducts have been formed. The one that we did notice and we did not notice with free chlorine is the formation of haloketones. So we didn't observe haloketones with free chlorine, but we did observe them with monochloramine. So that was an interesting trend that we observed with these ones. And so contrary to free chlorine, ozonated, UF ozonated monochloraminated water produced the highest disinfection byproducts in these experiments. This result suggests the ozonation increases the precursors that lead to a higher disinfection byproduct formation. So in the previous one, it eliminated if you use free chlorine, but in this case, it increased the formation of the precursors that led to these disinfection byproducts. And so the highest DBP class includes halonitromethane, followed by haloketones and iodo-THMs and halocetonitriles. And so if we compare the halocetonitriles between the UF and ozone and before ozone and after ozonation, we see an increase, a small increase of halocetonitrile formation, and this is not usual or observed in other studies. However, there was one particular study that observed a higher formation of halocetonitriles due to higher concentrations of bromide in the water. And so we did measure the bromide in the water, and we had about 40 micrograms per liter of bromide, which led to the increased formation of a brominated of bromochloroacetonitrile, and therefore we see the slight increase of halocetonitrial. The other thing that I wanted to really notice is the formation of iodo-THMs. So iodo-THMs, there was none before ozone, and then we see them after ozonation. It seems that that ozone produced 4 times more ITHMs in chloraminated than in chlorinated waters. Furthermore, it's significantly higher. And so we also tested for iodide in the waters, and we didn't find iodide in the waters. So what we suspect is that the iodide that produces this ITHM comes from the total organic iodine. So when I showed you the previous graph of what's contained in the wastewater, we did see about 4 ppb of total organic iodine. And so we believe that ozonation could have break down these compounds and after chloramination form these iodinated THMs. Especially we see a high concentration of iodoform in these waters. And finally, similar to other samples, the RO treated waters are the ones that has the least amount of DBPs from all the classes formed. Iodo-THMs was the largest DBP, and this was attributed to dichloroiodomethane. There was a few others that were also detected in these waters. Okay, and so we did also have some preliminary data by season. So the data I show you was from fall of 2019. And we did sample back in the summer initially, and as well as in the winter. But then due to the pandemic, we were able to sample further. However, we did see different trends by season. I'm not going to go in detail to all of them, but I just want to highlight a few of them. So we observed that dichloroacetonitrile seems to be DBP that comes up in most of all of the samples, as well as bromochloroacetonitrile. Dichloromethane was observed in fall and winter, but not in summer. And the very interesting is that we see these haloketones only forming in winter for HOCl treated waters. And so for chloraminated water, we also see dichloroacetonitrile form in all of the waters throughout the different processes, as well as dichloronitromethane. We did see a significant amount after ozonation in fall and winter. We did not observe this in the summer. We're not sure exactly why, but we need to further investigate this. We did observe haloketones in the fall and the summer, but we did not observe them in the winter. And iodo-THMs are present in all of the waters. And so this is just preliminary data, but I have a new student that's coming from overseas that's going to continue this work and also expand it into characterizing the organic matter or the compounds that are contained within these samples before they disinfect, to see what kind of precursors are forming these disinfection byproducts. It says conclusion. So halocetonitriles, dichloroacetonitrile was the largest forming halocetonitril in both disinfection methods. Bromochloroacetonitrile only formed in fall and winter, and also pre-ozonation followed by chloramine increased the halocetonitril formation, especially the brominated species, possibly due to high bromide levels. Dichloronitromethane was the largest forming halonitromethane in both treated waters. Dichloronitromethane formed at similar levels, regardless if it was treated with chlorine or combined chlorine. Halonitromethane is largely formed in UF ozonated effluents, as well as RO chlorinated waters. For haloketones, we observed that 1,1-dichloropropionone mainly formed in winter with chlorinated waters, and also it was formed in the summer and fall with chloraminated water. So the haloketones tend to appear, not appear in different, depending on which samples we treat them. Also, the 1,1-trichloropropionone and 1,3-dichloropropionone also form significantly in winter only. So the haloketones appear in some cases, sometimes it doesn't. And so for the iodo-THMs, the lowest forming group with free chlorine, it was had a high formation with combined chlorine, particularly with ozone combined chlorine and RO combined chlorine. And organic iodine precursors seem to promote the formation of iodo-THMs. And so with that, I would like to acknowledge the different partners and that supported this research, and as well as my research group here at Calgary. Thank you, and now I can take any questions.